all so much for coming. Yeah, I'm Matt. I'm really excited to talk to you all today about the evolution of evolution, or how the theory of evolution has changed over time. So, for a quick introduction about myself, I was born in Los Angeles, but I only lived there for a couple of months actually before I moved to where I grew up, Philadelphia. And I was always really big into biology. I was a big dinosaur person as a kid. And after I graduated high school, I went to do my undergrad at Cornell University, where I met my life partner. I got into a lot of watching movies, rock climbing, eating, and really got into that's where I really started to get into evolutionary biology and genetics. After graduating from Cornell, I spent a couple of years at Penn, where I worked in mice, and now I am at the University of Texas. I am a fifth-year PhD student, advised by my two wonderful advisors, Margaret Patrick and Arval Harpak, and this goofy group of lab mates. And I've really been learning a lot about evolution and evolutionary theory, and I'm excited to share some of what I've learned with you all today. So, to get started, I think that one of the things that really strikes people when we look around the world and the life around us is how incredible the diversity of life really is. You have animals, fungi, plants, bacteria, you see things that walk, things that swim, things that fly, and I think that one of the very natural questions that a lot of people have is, how did this incredible diversity of life arise? And the answer is, of course, evolution. I'm going to throw this quote up here from a very influential scientist, Theodosia Stojansky, who said that nothing in biology makes sense except in the light of evolution. And maybe some of you have heard this quote before. I think it really rings true. And today I'm going to try to zoom in on this word evolution and ask us ourselves what this word really means. So before I put out my definition of evolution, I want to ask you all in the audience, does anyone have any ideas that come to mind when you hear the word evolution? Any thoughts? Feel free to shout it out. Darwin, yeah.
Adapting.
Adapting, yeah. Fossils, yeah. Uh-huh, yeah. Change, yeah, nice. Cool, so I like a lot of these answers. Like I said, I was a dinosaur kid, so when I was first thinking about evolution, I was thinking about dinosaurs and how some animals seem to come and go over time. Some people mentioned adaptation and survival of the fittest. That's a very big part of evolution. Change, maybe some people think about changing from monkeys into people or something like that. No one said Pokemon, which is a little disappointing, but all of these, I think, maybe not the Pokemon part, but all of these I think are really important components of evolution. But the definition that I'm going to use today is this. Evolution is the heritable change in a population over time. This is a picture of a tree of life, which sort of shows the relationships between different organisms, how they evolved in relationship to each other. And I think that this diagram does a really good job of showing some of these key concepts of how things change over time and the evolutionary path that different organisms can take. So it's important to know that this definition didn't just come about in a vacuum. How we think about evolution has changed a lot over time. So here I'll pause and give a quick rundown of what I'm going to be talking about today. I'm going to start with the history of evolution and evolutionary theory. Then I'm going to talk a bit about how we think about evolution now, and then I'll conclude the talk with some ideas from current and future work. So to start with the history of evolution, I think it's important that we go back, like way back, like all the way back to some of the earliest civilizations. And really, a lot of ancient civilizations believed that life was created rather than evolved. We can see this in many of the creation stories all across the world. This is an example of one of them, The Dreaming, which is a creation story from some indigenous Australian groups. And this story says that humans and animals sort of arose from a void to populate the earth. But it's interesting to note that some cultures actually did believe that humans descended from animals. This is a picture of an Indri lemur, a type of monkey that lives in Madagascar. And the Malagasy people, who are the indigenous people of Madagascar, actually believe that Indri and humans are brothers and that they used to live together, but then the humans went to go live in the villages, the Indri went to go live in the forest, but they sort of share this ancestor. And really many cultures recognize that there was a relationship between living things. this idea was toyed with all the way back with the ancient Greeks, and was developed all throughout the Middle Ages. This is a picture of the Scala Naturae, which was one way that people thought of organizing all of the life, starting with plants at the bottom, then you had beasts, birds, humans, angels, and God at the top. Some people were thinking about more rigorously, scientifically defining organism relationships. Carl Linnaeus is one very famous name, and he really thought about specifically detailing species relationships. He actually is the founder of one of the still in use branches of biology, taxonomy, which is sort of the classification and grouping of organisms. And really, by at least the 1600s, people understood that organisms can change over time. A lot of these ideas came from things like animal breeding, so dog breeding, cattle breeding, pigeon breeding was really popular at this time. So people could tell that, if you took two fluffy dogs and you mated them together, you would get even fluffier puppies. And you could make these dogs fluffier and fluffier over time. Paleontology was also emerging as a sort of new field in science at this time, bringing in some ideas about how old the Earth actually is, species turnover. So again, this idea that some species seem to come and go over time. But creationism was still sort of the dominant thought really throughout the 1700s. All of these ideas were bubbling under the surface, but we still needed someone to come around and sort of propose more scientifically rigorous ideas for how this change was happening. The first person that I'm going to introduce that had some of these ideas was Jean-Baptiste Lamarck, who was a scientist in 1809 that proposed this idea for an inheritance of acquired traits. So what was Lamarck's main idea here? He thought that, so one of the things that he noticed is that individuals change over the course of their lifetime. We can see that we get bigger, for example, as we grow older. And he extended this idea to think about maybe individuals can intentionally change themselves to better fit their environment. The example that I'll give here is maybe these giraffes want to eat the leaves at the top of a tree, and so a giraffe might stretch out its own neck to try to reach the leaves, and then it can pass that stretched on neck to its children, and then those children will have slightly longer necks and stretch out their own necks, and so on and so forth, and eventually you would have a group of giraffes that could reach the leaves. This idea was very popular at the time, but we know that it doesn't really hold up now. For example, we know that if you get a tattoo, it doesn't mean that that tattoo gets passed on to your child. So a lot of Lamarck's ideas sort of don't hold water and don't really hold up to experimental scrutiny. And I should point out that poor Lamarck over here is often maligned as sort of the scientist that came up with a lot of ideas that we now consider wrong. But I'd like to point out, that this was only part of his ideas. This idea of acquired traits was very popular at the time, and he was still very foundational in a lot of how we think about evolution today. But that being said, we still need someone else to come around with a more modern idea that fits with our modern ideas of evolution. So of course, enter Charles Darwin, perhaps the most famous name in evolutionary biology, although at the time he looked more like this. Darwin was actually very well educated. He came from a wealthy family. He studied at college to be a naturalist, which is sort of the old school version of modern day biologists. He would go around and take draw pictures of birds and things like that. He was also a hobbyist fossil collector, a hobbyist geologist. And in 1831, he went on his now famous voyage on the ship, the HMS Beagle, whose goal was to go around to different places, survey the land, see what life they could find. And during this journey, which actually lasted 5 years, he stopped at this now famous island chain, the Galapagos. And there he observed variations on common forms. So this is a picture of some very famous picture that he drew of some birds, the Galapagos finches. And you can see that all of the birds look pretty similar to each other, but some of the birds have bigger heads, bigger beaks, some of the birds have slightly smaller heads and smaller beaks. So Darwin noticed that there was this variation in these birds and other species. And in 1859, he compiled his observations and his ideas in this extremely influential work on the origin of species. And in this work, he presented two core ideas: descent with modification and natural selection. This is the only figure that he put in the book. It's a tree that shows the relationships between different species, and I'll break down how these different ideas of his work. So the first one is descent with modification, which is the idea that children are slightly different from each other and from their parents. So this giraffe is maybe some height, and it have children that are also a slightly different height from the parents. And then his second idea was natural selection, which is the idea that some individuals are less able to survive and reproduce than others. This idea of survival of the fittest. And the important thing here is that which individuals survive is based on what traits they have. So for example, if again the leaves are at the top of the trees, maybe only the tall giraffes can reach those leaves, and so they're more likely to survive. The shorter giraffes are unable to reach the leaves and are more likely not to survive. And then Darwin is able to combine these ideas for his theory of evolution. So the tall giraffes would survive, they have children, those children, only the tall ones survive, and they have their own children, and so on and so forth. And eventually this population of giraffes should grow to be taller over time. So I should point out that Darwin didn't really have an explanation for where this variation came from. He also didn't have a good idea for how these traits are getting passed on. So for that, we're going to introduce the other big player, Mendel. So Mendel was a priest. He actually was also college educated. He studied philosophy and physics in college. And after deciding to be a priest, he joined an abbey and had a really nice garden, and there he was able to conduct these now famous pea plant experiments. So Mendel actually looked at several traits where individuals are either one of two things. So this is an example of some flowers that peas have, and Mendel noticed that the peas either had purple flowers or they had white flowers. And then he noticed that this trait gets passed on in very predictable patterns. So if he took the purple flower plants and bred them with the white flower plants, he would see all purple plants, and then if he took those and made more children, those children would be some purple, some white, in very predictable patterns and ratios. And so Mendel was able to deduce that there must be some underlying factor that determines the pea plant's flower color, and that gets passed on from parent to child. And this idea of something getting passed from parent to child is what we can now call heritability, or this factor is heritable. And so if we take these two scientists' ideas, we can come towards our definition that we're using now. Evolution is heritable, which we get from Mendel, this idea of something getting passed on, and a change over time, that idea comes from Darwin and his observations. And so now the question is, how do we combine Darwinian evolution and Mendelian genetics? And what is there to combine? So after these scientists put out their ideas, they actually created a bit of a rift in the scientific community, because people weren't sure which of these two scientists' ideas were better or more suited to what we see in nature. On the one hand, we have the biometricians who sort of followed Darwin's ideas, and they noticed that traits are continuous in a population, which means that you can have a whole range of different possible trait values. And so this matched more what we saw in nature, but their ideas for how this inheritance worked was unknown, and actually they believed something that Darwin also believed, this idea of blending inheritance, which is that the child is sort of the middle between both of the parents. We know that this doesn't really make sense, because if that were true, you couldn't have a child that was taller than both parents, for example. On the other side of the debate, we have the Mendelians, who were more interested in looking at discrete traits like Mendel looked at, which doesn't really fit as well with what we see with most traits in nature. But they had more well-defined laws of inheritance and how these traits get passed down. So there was this conflict that needed to be resolved, and these three scientists were really at the forefront of resolving this conflict. Haldane, Wright, and Fisher, they were able to bring in ideas with statistics, mathematical modeling, and really came up with a framework for specifically how population might change genetically over time. And so this idea that continuous traits can actually follow discrete Mendelian inheritance laws is called the modern synthesis. And now we're really close to how we think about evolution today. But you might notice that I've been talking a lot about genetics. We're missing a very key player in all of this evolution talk, and that is, of course, DNA, deoxyribonucleic acid. This molecule was first discovered in 1869 by Friedrich Miescher. He was a physician. And after he discovered DNA, it actually took a really long time for people to realize what DNA actually did in our cells and in our bodies. And it wasn't until decades later, in the 1930s and 40s, culminating with this paper from Avery, McFaul, and McCartney, that demonstrated that DNA is actually what is getting passed on from parent to child. This is a picture of the actual cells that they were studying. In 1953, of course, we have the very famous results from Watson and Crick, along with work and data from Rosalind Franklin and others. And they proposed this double helix structure of DNA. And now we really sort of had a revolution in how we think about evolution. Instead of just thinking about traits getting passed on, we were now able to think about DNA and genes getting passed down from one generation to the next. And this really opened the floodgates for us to think more about how important DNA really is. In 1977, Frederick Sanger and colleagues developed Sanger sequencing, which is one of the first more modern techniques for sequencing DNA, meaning getting the string of letters. In the 1990s, there was continued development of Sanger's ideas in what we call next generation high throughput sequencing, which really used better computers, faster processing speeds, things like that, to allow us to sequence much longer DNA sequences and many more DNA sequences. In 2000, we had the first whole human genome, meaning all of the DNA in a person. We could sequence that. That was sequenced by the Whole Genome Project, the Human Genome Project. And throughout the 2010s and continuing now to the 2020s, there's been continued improvements to whole genome sequencing. This figure shows that the price to sequence a single human's genome has decreased dramatically. In 2000, when they first sequenced it, it cost $100 million, and now it costs like $100 or less. And you can see that actually the cost to sequence has outpaced our expectations. And so all of these scientists, Darwin, Mendel, the fathers of the modern synthesis, all of the scientists that worked on DNA, they all contributed to now this definition that we use today. Evolution is heritable change in a population. So I will now get into how we think about evolution now. But before I go, I'm going to mention a couple more points about this definition. The first is that when we're talking about evolution, we're really talking about populations changing. So again, Pokemon, for those of you who don't know, the idea is that you have one individual that makes big jumps from one stage to the next to get more powerful. And this isn't really how evolution works, because evolution works on whole populations, not single individuals. And also, evolution doesn't occur in huge leaps. It mostly occurs in very small little steps over time. And when I say over time, that actually, what we mean by over time actually can change based on which organisms we're looking at. Humans, we measure a generation in decades, but some organisms, their generation time can be minutes or hours. And a lot of that has to do with how big the organism is. And so when we talk about over time, a lot of times we're talking about how over the course of many generations, rather than over the course of some number of years, for example. So how do we think about evolution now? There are sort of two main branches of evolution, evolutionary research nowadays, specifically evolutionary genetics. That's population genetics and quantitative genetics. Population genetics focuses on evolutionary forces causing a change, and quantitative genetics is how traits change over time. So I'll talk about both of those now, starting with population genetics. So again, population genetics is interested in these evolutionary forces that contribute to change. The forces that I'm going to be talking about are mutation, selection, drift, and gene flow. And I'll point out that Darwin had really pretty modern and good ideas about how selection worked, but he didn't know about all of these other forces. So we'll talk about how each of these work now. Before I get into that, a very quick refresher about how DNA works. So DNA is this molecule that's composed of 4 bases, C, T, G, and A. And in our bodies, those bases exist in a big long chain. And so we can compress that chain down into just thinking about the sequence of the letters representing those molecules. When we're talking about a genome, we're talking about the entire DNA sequence in one of the cells in our body. And when we talk about a gene, we're talking about one specific section of the genome. So you could imagine that this little sequence here represents a gene. And the important thing is that this DNA sequence actually gives us instructions about how our body should look and function. So the first important force for the evolution of these sequences is mutation, which is the source of variation in a population. In genetic terms, you can think of this as a change to the DNA sequence. Perhaps you change the C to a T, for example. It doesn't have to just be single letter changes. You can also have insertions of big chunks of DNA. You can also have deletions of large chunks. And the important thing is that whatever change occurs, it needs to have some effect on what the individual looks like. So for example, let's say we have this group of lizards. Maybe the lizards that have a C are green, and the lizards that have a T are brown, something like that. And I'll point out that mutations are really just random DNA typos, essentially. So there's no rhyme or reason as to which type of mutation occurs, which letters get changed, and what letters they get changed into. All of this is completely random, and this variation just sort of occurs spontaneously. And population genetics is really interested in how common are these different versions of DNA, or what we call alleles, and do those frequencies of these alleles change over time. So as a starting point, let's imagine that 50% of our lizards have a C and 50% of our lizards have a T. So if we're thinking about how these different versions of DNA are changing, we have to think about how or why. And one reason could be through selection, which is when one version becomes more common because it's advantageous. When I say advantageous, what do I mean? Let's imagine that these lizards live in the grass. The green lizards are going to maybe blend in better. They're not going to be seen by predators as much, and so being green would be advantageous, and correspondingly, the C allele would be advantageous. And conversely, brown lizards sort of stick out on the grass, and so they would be disadvantageous, and the T allele would be disadvantageous. And so this advantage is actually going to lead to a change in the frequencies of these different versions of DNA. And actually, we should see that the more individuals over time will start to be green and have a C version of the DNA, and fewer individuals should have a T. In this case, the C went up from 50% to 80%, and the T went to 20%. And actually, if selection is the only thing occurring in a population, we should see that eventually every single individual will have a C, and no individuals will have a T eventually. But know that selection isn't the only evolutionary force occurring. We just talked about mutation, which is always acting in the background. Another very important force, and we're only continuing to learn just how important it is drift, which is this idea of random chance that can impact changes in frequency. So for this, I'm going to use another example. Let's imagine that there's a disease that's affecting these lizards, and this disease doesn't care if you're a green lizard or a brown lizard or a pink lizard or a rainbow lizard. It's going to infect all of the lizards equally, and eventually 50% of the lizards in the population will die. So it's possible that this will cause no change in the frequencies of the green and brown lizards just by random chance. But if it just so happens that all of the lizards that die are green, then we would see that the proportion of brown lizards in the population actually goes up, again, just completely by chance, depending on which individuals get killed by this disease. And so we can see that this would actually lead to a change in the versions of the DNA as well. The C would go down from 80% to 60%, and the T would go up to 40%. Even though we just said that T is actually disadvantageous, it still goes up just due to the random effects of this disease and the randomness of genetic drift. The last evolutionary force I'm going to talk about is gene flow, which is this idea of movement of individuals from one population to another. So we could imagine that we have two small populations of lizards, and maybe there are lizards constantly moving from one population to another, you could imagine that this would also affect how many brown and green lizards exist in one of these subpopulations at any given time. And really all of these four forces are interacting constantly to drive genetic changes in populations. Again, Darwin knew about selection, and that selection oftentimes gets sort of the spotlight when people think about evolution. But really all of these forces are potentially equally or even more important depending on the specific populations that we're looking at. So that was a brief overview of population genetics. I said the other sort of subfield that I'll think about is quantitative genetics. And to start my this section of the talk, I'm going to mention that Mendel actually got super lucky with the traits that he observed. I mentioned before that he looked at flower color, these purple or white flowers. He also looked at things like the shape of the seed of the peas and the color of the peas and the colors and shapes of the pods, things like that. But you'll notice that all of these traits that he looked at were sort of either or. You're one thing or the other. And we know that that's not really how traits work in nature. Again, I said before that most traits that we see are actually continuous, meaning you can have any number of different possible traits rather than discrete, which means you're one thing or the other. This is a picture of human height, which is a very famous continuous trait in humans and really in most organisms. You can see, and I'm sure that you can probably think of some people who are very short, some people who are very tall, and people of all heights in the middle. So that was one thing that Mendel wasn't able to fully account for. The other thing is that all of the traits that he looked at were sort of affected by one gene. But we know now that actually most traits appear to be affected by more than one gene, or what we call polygenic. This is a picture of a very popular type of analysis nowadays called a genome-wide association study, or GWAS. And the idea here is that we're going to scan the entire genomes of all of the different places in our DNA, and we're going to look for genes that are associated with some trait. In this case, we're looking for genes associated with height. And you can see that these researchers found a ton of different genes that appear to be associated with how tall someone is. And the study was put out in 2009, and since then we've only found more and more genes that appear to be associated with height and all human traits. So these were some of the things that Mendel wasn't really able to observe or account for, but that doesn't mean that we have to toss his ideas out the window. To better understand how, I'll first mention that many organisms, including humans, have multiple copies of each gene, one that we get from a mother and one that we get from a father. You can imagine that you have some gene here, the B gene, and you can have two versions of this gene, a big version or a little version. And if you take this idea, you can see that continuous traits and polygenicity are actually still compatible with Mendel's ideas. If you happen to get two of the little versions of this B gene, maybe you're really short. If you happen to get two big versions, you're tall. And if you have one of each, you might be somewhere in the middle. And if you have two genes affecting a trait, you can actually get more variations. So if you have an A and a B gene, and you have little versions of both of those, you might be really short. If you have all big versions of both the A and the B gene, you might be really tall, and all of the different combinations in the middle might lead to intermediate heights. And the more genes you have affecting a trait, the more continuous that trait can appear. And so really, most traits exist in distributions in the population, by which I mean some individuals have one trait, some individuals have another trait, and you have individuals at different traits in the middle. This is an example of one type of distribution, or normal distribution, which is where you have some individuals that are at the extremes, but most individuals are somewhere in the middle. This is, of course, not the only way that distributions can look. You can also have something like a bimodal distribution, which is where you have more individuals at the extremes and fewer individuals in the middle. A neat example of this is these weaver ants, where within one colony you actually have some ants that are really big and some ants that are really small. And the really big ants sort of go out and help forage, and the small ants sort of stay in the tunnels and help manage the tunnels. And evolutionary quantitative genetics is really interested in how do these trait distributions change over time. One way that you can imagine they would change is through something called directional selection, which is where a trait changes towards one direction. In this case, maybe it's better to be a tall giraffe, so the population is going to change to become taller over time. One cool example of this is these pepper moths, which initially were all white to be able to blend into these trees that they lived on, which had white bark. But in the 1800s, there was the Industrial Revolution, and actually all of these factories were putting soot into the air, and the soot covered the bark of the trees and turned the barks black. And so these moths very quickly evolved to become black themselves, so they could blend in with these new soot-covered trees. And this is an example of really rapid directional selection, where the moths went from being mostly white to mostly black. Another cool example is these garter snakes that eat toxic newts, and these actually have a really cool evolutionary cycle going on, where the newts are constantly trying to evolve to become more toxic to avoid getting eaten by the snakes, and the snakes are constantly evolving to try to become more toxic resistant so they can keep eating the newts. Another way that a trait distribution could change is through stabilizing selection, which is where the trait distribution becomes more narrow. Here, maybe it's bad to be in one of the more extreme heights of giraffes, so all of the giraffes sort of coalesce towards being a middle height. One cool example of this is robin egg number. So robins almost always lay exactly 4 eggs. And the reason for this is that if you lay 5 eggs, there might not be enough food and resources to go around, and so the chicks are more likely to die. But if you lay 3 eggs, you're sort of not maximizing the potential number of eggs that you could lay. So almost all robins actually lay exactly 4 eggs. Another example is these cacti, which have very specific spine densities. If you have too few spines, then you might be more likely to have spines. You might have too many hiding places for parasites and things like that. So these robins and these cacti have evolved towards very specific intermediate trait values. And then the last way that I'll talk about a trait distribution changing is through disruptive selection, which is where a trait distribution becomes more spread out over time. So here the extreme values are better than the values in the middle. This could occur. One example of this is those weaver ants that I mentioned before. Another is going all the way back to Darwin's finches. On the Galapagos Islands, food exists sometimes in these really big seeds, and sometimes in these little holes as like nectar or insects, things like that. And so some of the birds have really big beaks to be able to eat the big seeds. Some of the birds have really small beaks to be able to reach into the little holes, but not that many birds have beaks somewhere in the middle, because then they wouldn't be able to eat either food source that way. And one of the important things is that we can actually combine these ideas from both population genetics and quantitative genetics to understand how a population is changing over time. So we might be able to see that this population is changing in how tall the individuals are. And then we can also think about which of the evolutionary forces are causing or contributing to that change. And then we can further think about how are the underlying genetics and the frequencies of the different versions of DNA changing over time. And so really, I mentioned that these are sort of 2 subfields. Really, they're actually 2 sides of the same coin, and most researchers nowadays think about both of these aspects of genetics and evolution when they study evolution now. So what are some of the big takeaways that I want us to come away from this section about how we study evolution nowadays? The first is that big changes occur in small steps. So even something like a whale evolved through a series of very small changes over long periods of time. For example, what might have started as a paw, the fingers got longer and longer and turned more and more fin-like as the whale became more and more well adapted to living in the water. And really everything from bacteria all the way up to the biggest animals and tallest trees, all of those different forms came about through very small little changes that accumulated and built up over time. Another takeaway is that we don't always change, we don't change to fit the environment, we change randomly, and the environment impacts which changes persist. So again, this is yet another knock on our poor maligned friend Lamarck over here, because Lamarck's idea was sort of that individuals can intentionally and deliberately change themselves to fit their environment. But now we know that really changes sort of happen in the population completely at random, and it's through selection that some of those, through selection, drift, gene flow, all of those forces that some of these changes persist in the populations. And the final takeaway that I'll put out there is that evolution doesn't have an end goal. Selection just sort of looks at what challenges are present in the environment and looks for quick fixes to those challenges. One example I'll put out is this sickle cell trait, which in populations of humans which are heavily affected by malaria, it's actually really good to have a copy of this sickle cell gene because it provides protection from the malaria parasite. But for humans that are living away from areas that are heavily impacted by malaria, the sickle cell is actually disadvantageous because if you get two copies, then you have sickle cell disease, which has a lot of really negative health effects. So this is an example of a trait that has pros in one environment, but sort of cons in another. And there's no sort of higher design of like moving towards a perfect organism. Selection is just putting changes, looking at what challenges there are in the environment and looking for fixes. And so the definition of evolution that we use continues to update and change over time, and we're only continuing to learn more about how evolution works. So now I'll take a moment to talk about some of the current and future work going on in the field. Of course, I'll start out by pointing that, of course, evolution is still occurring in all organisms today. A lot of this evolution is actually occurring in response to man-made changes in the environment. These are examples of some fishes that have developed pollutant resistance because the waterways that they live in are getting chemicals are getting dumped into them from factories, things like that. This is an example of a plant where some of these plants have developed herbicide resistance in order to survive some of the like weed killers and things like that used by people in agriculture. Another interesting modern example is that you're probably all familiar with is COVID-19, where we can see that which strains are most common in a population have been changing over time. These different colored bars represent different strains of the virus. Going from left to right is different months of the year. And you can see that the proportion of which strain is most common has changed a lot over the past year, indicating that this virus has been evolving very rapidly to change how it affects people, how severe it is, how it gets transmitted, things like that. Humans, of course, are no exception. Humans are still evolving as well. There's a lot of evidence for both older and more recent human evolution. This map is a picture of lactose tolerance, which we believe evolved extremely rapidly. It's changed dairy into their diets, which overlaps with the higher lactose tolerance, and some cultures didn't, and those would be the lower lactose tolerance. There's also been some evidence that the age of menopause has been increasing over the last several decades, and maybe is even continuing to increase now. I should point out that traits like this, especially like menopause, there's a lot of different factors that go into it, and we're still sort of trying to understand how this trait is changing, what are the different factors, genetic and environmental, that contribute to it. Of course, how we study evolution is itself changing. Ancient DNA, for example, is becoming increasingly viable for research. This is a picture of some results from a study that came out where they actually were able to sequence a whole Neanderthal genome using much updated DNA extraction techniques, DNA sequencing techniques to be able to sequence DNA way older than we could ever look at before. They were able to show how different Neanderthals and humans are from chimps, and also that Neanderthals are somewhat different, but actually not that different from humans. And this work was actually awarded the Nobel Prize to Svante Pääbo, who was the head investigator. And I believe he's the only evolutionary geneticist to ever be awarded a Nobel Prize. Another really interesting and emerging field is epigenetics, which is this idea that not only our DNA sequences are impacting what we look like, but also modifications to the DNA. And the interesting thing is that these modifications can actually be due to environmental factors like diet, chemical exposure, or exercise. And these modifications get added on top of the DNA rather than being a change to the DNA sequence itself. And so you might notice that, you know, this might be an example. Sorry, I should point out also that these modifications can get passed on. There's some evidence that these modifications can get passed on from one generation to the next. And so this actually seems like maybe it's an example of a trait acquired from the environment that gets passed on. So maybe this can help redeem our friend Lamarck here, who has been sitting on the sidelines for so long. But it's pretty contentious how much this actually sort of props up Lamarck's ideas, and it's not clear how long these epigenetic changes persist in the population and how they get transmitted. We're also becoming increasingly aware of just how much our environment and genetics intertwine. This is a figure from a paper that was just put out last year from actually some of my lab mates and colleagues, where they showed that it's not just our genetics that get passed on from one generation to the next. It's also things like wealth and education and connections. And when we're thinking about this idea of nature versus nurture, there's actually, it actually turns out that it's nature and nurture and the interaction between the two that determines what we look like and what traits we have. So, sorry. And so really there's sort of this interplay constantly going on between our genetic ancestry and our environment. Cool example of this is with asthma incidents in Black communities, where it was some studies had shown that there was a correlation between asthma and some Black ancestry genetic markers in Black American individuals. But it turns out that this was sort of conflated by Black people more likely living in urban environments where there's higher exposure to things like air pollution. And so really there's a constant interplay between genetic ancestry and environment that we have to be aware of. And we really need to be careful about making broad statements about how genetic affects human traits in general. This book, The Bell Curve, was published in 1994, and it suggested that things like intelligence, social status are strongly affected by genetics and associated with race and ethnic background. And this book was really widely criticized by a lot of scientists since it's been published. And it serves as a sort of important reminder about the possible pitfalls that we can encounter when studying and presenting on evolution. And importantly, this book suggested some really controversial policy ideas based on its conclusions. And so evolution research can really have very strong real-world consequences for how it's applied in our society and in our world. So although we need to be careful about how we interpret and present our findings, I will suggest that evolution research is, of course, still critically important, and perhaps even more important than ever, to help us disentangle some of these challenges that I just mentioned. And really everything from disease to conservation to human society and culture, evolution research is critical for understanding this and our world and ourselves. And so I'll leave us again with this quote: "Nothing in biology makes sense except in the light of evolution." And really, it's only through evolution that we can understand biology and the world around us, our place in it, and how we can best preserve and protect our planet and ourselves. Thank you very much for your attention, and I will now take any questions from you. Yeah, so I think we have a mic that we'll pass around for any questions. Yes.
Is this Mark?
Hi, that was a great talk. Thank you. My question, since you mentioned you worked on CRISPR before, and now we're utilizing CRISPR to potentially change the genetic makeup of humans. So how would that affect evolution if we're now potentially changing deleterious effects that now would not, or prevent that individual or that set of individuals in the population for passing, so that they wouldn't pass those traits?
Yeah. So I actually have a supplementary slide about this. So yeah, the question was about CRISPR. So it was about CRISPR and this idea of how might genetic modification affect human evolution in the future. So for those who don't know, CRISPR is this really neat technology that emerged in the last decade or so that allows us to change any sequence anywhere in the genome. And it has a lot of really impactful potential effects on evolution, genetics, and yeah. So one of the big things that people have been thinking about with CRISPR is sort of this idea of, can we make our babies have like any sequence that we want? Can we control what traits we give them? And the answer is that yes. So theoretically, yes, we could. There's been a lot of research that's been going into this, but it's very, very controversial for a few different reasons. So to more directly answer your question, I don't think that we're anywhere close to being in a position where CRISPR will directly influence human evolution in the near future. And the reason that I say that is that we just don't understand well enough how all of the different components of our genome work and interact to know when we change one thing, is that going to change something else that we didn't anticipate. CRISPR technology itself is still not a perfect science, and there's still a lot of like issues that can arise with the babies not having, like, not all of their cells or get CRISPR modified, for example, things like that. And of course, there's the ethical concerns as well, where people are pretty hesitant to just allow us to modify whatever we want. This picture here is this Chinese scientist who actually went and CRISPR edited a couple babies in China, and they were born, they had CRISPR editing, and he was really, really widely criticized for doing this. He was imprisoned, actually, as a result, and it's still unclear exactly. So he was trying to help make the babies less susceptible to HIV, I believe, but it's actually pretty unclear, like, what other side effects might occur in those children. So I think that CRISPR and DNA editing definitely have the potential to alter human evolution, but I think that we're not really, the technology isn't there yet, and I think that people in the scientific community recognize that there's a really big risk with it. I also put up this picture of this movie Gattaca, maybe some of you have seen it, where this society exists in the future. Some people are genetically modified and some people aren't, and this leads to like really big, like social stratification and things like that. I do think that there needs to be maybe like people are thinking about like legislation and things like that, and hopefully like in preparation for and to prevent some of these issues from arising. But yeah, so I think that CRISPR is very powerful, but its effects maybe won't be felt soon. Did that answer your question? Mm-hmm. Any other questions?
We have one here.
Yes.
Hi, so it's on. Well, I don't know. Okay. Anyways, let's not talk loud. I wanted to ask you to elaborate a little bit more like on epigenetics. I remember like 12, 15 years ago talking with a friend who worked in neonatal care and epigenetics, and vaguely what I remember is just this idea about how, like you said with Lamarck, it's like there's certain, like in our genome, there's traits that are existing dormant and that can be turned on or turned off depending from anything from like diet, exercise, trauma, like psychological impact of like early childhood or PTSD in life that then do get passed down to our children. Even like, drug dependency is like a very classic example that's been studied a lot. And so I was just wondering, like, if you could help explain or tell us a little more about that, just like, one could say that obviously if you want your babies to be the healthiest version, you want to live a good, healthy life to hopefully activate those genes that you'll then pass on. But I don't know how plausible that actually is, or if that's kind of just my understanding of that might be right.
So So I'll quickly expand a bit on what epigenetics actually is to answer that first question, because I really glossed over it pretty quickly here. One element of DNA that I didn't mention, so I said that DNA sort of provides instructions for our body, and the way that it does that is that DNA provides instructions for making proteins. And epigenetics is really this idea of how much of a protein is being made by our DNA. We can actually change how much of different proteins are being made based on not just DNA sequences, but also like how easy is it for our cells machinery to get in and access different parts of the DNA and things like that. So a lot of that is what epigenetics is concerned with, how accessible and sort of like what shape is the DNA taking, and how does that affect how much protein is being made from that DNA. So to get to the second part of how is this passed on, so a lot of epigenetics is through these things called like genetic markers or DNA markers, DNA epigenetic modifications, which are little molecules that get added on top of the DNA sequence. And there is some evidence to suggest that these can get passed on, like you were saying, from parent to child, but it's not clear exactly how this transmission happens and how permanent it is. So a lot of research has suggested that so first of all, not all of the modifications that occur get passed on. And it's not clear which ones get, like why certain ones get chosen to get passed on. And it's also not clear how long these modifications persist for over the course of multiple generations. So even if you pass something on to your child, it might not continue to get passed on even to like your grandchild, for example. So yes, there's definitely a huge impact in terms of your environment can definitely change your traits, not just because of like your lifestyle, but actually on a genetic level. It seems like your environment can affect your genome, but it's not clear exactly how much that affects.
Kind of that nature-nurture balance.
Yeah, so the nature and nurture is sort of a constant. There's always a bit of both going on. And really there's a lot of interaction between nature and nurture as well. It's not just genetics or environment, it's genetics times environment sometimes. Did that answer your question? Cool. Any other questions? Yes. Sorry, say that again.
On average, how long does it take to process?
In humans, So generally, evolution takes place over many generations. But it sort of depends on a lot of different factors. For example, if we're looking at something that's like getting selected, how strong selection is can affect how quickly evolution might occur. The size of the population that you're looking at could affect how quickly evolution occurs. In humans, I would say probably, I'd have to think about an exact number, but I think that there's a lot of different factors that go into it, so that's pretty like trait dependent, I would say. But we can also, so another aspect of evolution that we can look at is, actually, I'll just leave it there. But I would say that it's trait dependent. Some of the faster traits you could probably observe over the course of just a couple generations or a few generations. Some of the traits where selection or population might be changing very slowly, maybe many generations, like hundreds, thousands of generations.
Oh, maybe we can do one small question, just the last one.
Yeah.
And I'm happy to stick around afterwards if you have
great.
questions.
Thank you.
Maybe piggybacking a little bit on the epigenetics, since that's, like you mentioned, protein expression. but focusing on the step before, like the smaller RNAs, like microRNAs and siRNAs, because they also modulate gene expression. It's been a long time since I studied this, so I'm not sure. Has it been studied, their impact in evolution, and are they being passed on, and how are they being?
Yeah, so, yeah, this is a good question. So again, even my more detailed explanation was a bit simplified from before. So the path from the instructions encoded in the DNA to what we look like is through DNA gets turned into RNA, and then, or not turned into, but it's used as a template for RNA, and then that RNA is used as a template for protein. And so really what epigenetic modifications do is change how much RNA is produced, and then that in turn affects how much protein is produced. To your question about different types of RNA, so DNA, so another thing that I sort of simplified, DNA doesn't just have genes in it. It also, by which I mean it doesn't just have things that make proteins. There's actually tons of our genome, really long stretches of our genome that don't make any proteins. Some stretches of our genome seem to not do anything at all. Some of them make these other types of RNA that are really important, like tRNA, which help like make the proteins that that DNA creates. So all of those different factors can evolve. Anything that's coded for in the DNA can evolve, including these non-coding regions, like the regions that turn into tRNA. Anything that's RNA can evolve, and can evolve through epigenetics as well, because anything that needs to be made from using DNA as a template could evolve through either DNA evolution or through epigenetic evolution.
But is there any impact? Have we looked at how the siRNAs and the microRNAs are impacting evolution itself?
MicroRNAs, siRNAs, these also have to do with like the final protein product. They help with like, as far as I know, they help with also with like expression, protein processing, things like that. So yeah, and those are also coded for by the DNA. So they can evolve. And for example, a lot of changes in our genome are. Sorry, let me back up. So a lot of changes in our genome don't just affect what protein is being made, but how much of the protein is being made through things like these microRNAs, or through things like promoter regions or enhancers, or things like that. So there's a ton of different ways that evolution can occur, either because a protein itself is being changed, how much of the protein is being changed. There's a lot of different pathways that can take, including epigenetic pathways, including microRNA pathways. So did that answer your question? Yeah. In summary, there's a ton of different things that can occur to lead to genetic changes, to evolutionary changes, and a lot of different pathways that can affect. Any other questions? Thank you so much for your attention.
All right, thank you all for coming, and thank you, Matt, for a wonderful talk. Just one quick announcement for those of you that are still here. If you are interested in doing any other activities that are part of the Texas Science Festival, or interested in coming back here, there is an open house here this Saturday from 10 to 3. You can check out the property, learn more about some of the research that's actively happening here, and also check out some cool parts of the UT's insect collection and some different tabling and other activities. So I have 2 flyers up here if you want to take a picture of one. There's also a QR code that'll link you to the website so you can get more information if that's something you're interested in. But again, thank you so much, Matt. That was a wonderful talk. And if you're able and willing to, would you mind helping us pick up the chair that you are sitting on, just to make it a little easier for us